For joining us today. We are going to get started for today's webinar. And essentially, we are going to start off with some updates from the Knowledge Portal, and then we will go into our speaker presentation, and we can go from there. If you have any questions that you would like to ask, or if you have any comments, please feel free to utilize both the chat or the question and answer. We will be monitoring throughout the entire session, so we're really eager for you to, ask your questions and participate along the way. So with that being said, I'll get started on today's updates. So starting off, I just want to highlight two of our conferences that we'll be attending as the Knowledge Portal Network. The first is the American Diabetes Association, which will be in Chicago in June, and will also be at the American Society of Human Genetics in October in Boston. So these are some of our regular conferences that we love to attend. We love to see people and chat with you all about the different science and updates that we have. So if you're going to be at either of these, we'd love to see you there. Once we get closer to the date, we'll have some more information regarding the booth and different events that we'll have at both of these sessions. Another webinar series that we host is the Common Fund Data Ecosystem Public Webinar Series. Our next session is going to be Friday, April 25th from 1 p.m. to 2 p.m. Eastern. And we will have Dr. Alyssa Dillman from the Training Center share hackathons with impact, data-driven projects with the CFDE. We hope you'll be able to join us for that as well. Just a couple of the other network meetings that we have for 2025. Upcoming, we have May 8th for a focus group and June 12th for our webinar. And now I'll hand it over to Matthew Sampson, who is going to share with us discovering the genomic drivers of nephrotic syndrome towards improved kidney health. So with that, I will stop sharing and I will hand it off to Matt.
Great. Thanks so much. Can you all see me?
Yes.
Great. Okay. And then I'm going to try to share my PowerPoint. Let's see. Microsoft PowerPoint. Aha. Okay. All right. So looks good or what?
Yes, that looks great. Thanks, Matt.
Okay. Well, thanks so much, Mackenzie, for the intro. It's really great to be here to share a little bit about nephrotic syndrome and talk about our work with the Knowledge Portal team towards understanding or helping us understand nephrotic syndrome. So, I'm going to run through this. I want to contextualize the known genetic causes and contributors of nephrotic syndrome with the framework of genetic architecture. And I want to share our collaborative NIDDK RC2 funded project on nephrotic syndrome genetics, in which Mackenzie and the team are key contributors. So basically, and this is the work really of this RC2 project, really the work of kidney genomics is the work of many folks, but the core team that I'm working with is Drs. Martin Pollack, Simone Sanakerke, and Freedom Hildebrandt. In particular, Simone Sanakerke, who is on this webinar as well, associate professor at Columbia University, is really driving a lot of this work. So, I've been caring about nephrotic syndrome for a long time. This is one of my patients that I saw in during my fellowship, and he had swelling around his eyes and in his belly because his kidneys were losing a large amount of protein into the urine. And over the years, he's suffered from this condition. Nephrotic syndrome is a chronic disease, and at times he goes into remission of his proteinuria, so his swelling goes away. And then over the years, it relapses as well. And, he's 18 years old, and his life for himself and his family has suffered quite a bit because of it. What we know about nephrotic syndrome from an anatomical perspective is it's a disease of the glomerulus, which is the kidney's filtering units. This is a kidney and cut in half. We each have two kidneys usually, and we have about 1 million of these filtering units in each kidney, and blood and blood's components, which include cells and protein, comes into the filtering units. The good stuff stays in and goes back into the bloodstream. The bad stuff like waste and goes out into the urine. But nephrotic syndrome, there's a defect in this barrier and there's a loss of protein into the urine. And, I saw pictures like this during my my medical school training, and this really hit home for me, the damage that nephrotic syndrome causes. So this is a scanning electron microscopy, like you're floating in the urinary space, staring down the glomerular capillaries and seeing these beautiful podocytes, these epithelial cells, interdigitating and giving a hug to the endothelial cells. That's providing a size and charge barrier to keep the good stuff in and let the good stuff out. However, during nephrotic syndrome, what we see is a destruction of, or an injury to these podocytes in the glomerulus, leading to a flattening of these podocytes, a loss of these beautiful interdigitations. And sometimes we can get this better with therapies. Other times this inexorably progresses to end-stage kidney disease, chronic kidney disease. And the issue is like, we can look at this on microscopy and say what we see, but we don't know why this happens, and because we don't have a great molecular understanding of why this happens, we don't have a great idea of what we can do about it. So this is pretty bothersome to our whole field, and the challenges that frustrate us are that these clinical classifications are descriptive and they're non-specific. They are classified. We base patients on their response to steroids, so we call a disease steroid-sensitive nephrotic syndrome or steroid-resistant nephrotic syndrome. If there are minimal changes on light microscopy with a kidney biopsy, we call them minimal change disease. And if there are focal areas of scarring, we call it focal segmental glomerular sclerosis. These are all descriptive and don't give a molecular understanding into the disease, which could give us an entree in terms of predictions and better care as these molecular markers as biomarkers. Or these molecular markers can be beacons towards understanding biology. The other issue is that we can treat nephrotic syndrome, and sometimes we can achieve complete remission of the proteinuria, but the treatments themselves cause lots of side effects. So we've cared about this for a long time in our field, and what myself and Simone and a number of other folks have been doing is attacking this problem through genomic discovery, which I've sort of nicknamed kidneyomics. And it's the idea that if we can understand the genetic basis of nephrotic syndrome, we can discover disease-causing or contributing variants. We could illuminate the clinical impact for those harboring the disease variants. So this is, genetic drivers as a biomarker. We could deliver novel insights regarding nephrotic syndrome biology to identify new targets for medicines. And ultimately, lots of people are trying to develop drugs that are genetically inspired. And alternatively, we can once we find genetically, drugs against specific genetic signatures, if we know our patients' genetic signatures, we can select them for clinical trials more effectively. And ultimately, there's so many phenotypes that are doing this, and we can just add ourselves in the nephrotic syndrome to the list about delivering genomically informed care. So I just want to give you guys a brief highlight of how we discovered the genetic architecture of nephrotic syndrome. Like many other phenotypes, the history of genetic discovery typically has started in families where they're multiply affected individuals, which was the only way we could be empowered to discover genes. But alternatively, now we can study genetics through population-based approaches, and it's been successful in both ways. So this is, nephrotic syndrome as a Mendelian disease historically, nephrotic syndrome as a complex disease, GWAS burden analyses, etc. So what we all aim to do collectively is we do not have the luxury of only taking families or only taking populations. It's a rare disease, and we have to make the most use of every single patient that comes our way. So whether it's through families or through populations, we are aiming to discover, novel nephrotic syndrome associated genetic variants, which is always kind of cool to discover something new. But we already have a lot of genetic variants, so it's just as important to us to discover known genetic variants in patients who have them. And if we can do that, we can look at clinical associations, and we can take the mechanism to medicines approach. So I'm sure most of you have seen this figure before, which contextualizes the genetic architecture of disease on two axes: allele frequency ranging from rare variants that hardly ever appear in the population to those that are common, and effect size indicating the likelihood that someone will have the disease if they harbor the variant. And we can go from Mendelian disease that are very rare or novel, which have a huge effect, or common variants which are present frequently in the population but have small effect sizes. And I'm going to show you stories in nephrotic syndrome that apply in each of these quadrants. And again, we use families and populations, and families typically are the rare ones with big effect size. Populations have historically been others, but as we'll show you, we can do some things with populations for rare variants as well. So let's start with the architecture of rare alleles causing Mendelian disease, and that is the clinical correlate of that is steroid-resistant nephrotic syndrome and FSGS. So nephrotic syndrome is a rare Mendelian disease over the past 30 years, has uncovered about 60 genes that collectively, might explain maybe 15% of all patients in the world with steroid-resistant nephrotic syndrome. And these genetic variants, the gene products have really localized to the podocyte. So this is the map of the podocyte by, again, one of our RC2 collaborators, Dr. Friedhelm Hildebrandt, showing that these genes that are broken through a mutation can kind of converge around biological nodes, whether it's CoQ10 biosynthesis, actin regulation, the slit diaphragm. So this is important, and what we've been able to see is that the prevalence of Mendelian forms of SRNS, FSGS differ by location around the world. In places where there's a high amount of consanguinity, you maybe perhaps 45% of cases would be Mendelian. However, in places where probably most of us are on this call from, where there's not as much consanguinity and people from multiple genetic ancestries come together, aka different races, whether it's Brazil or the US, maybe it's about 8 to 15%. And then it also depends on whether we're also looking at congenital nephrotic syndrome. So the younger you are, nephrotic syndrome in the first 3 months of life is often is like 60-70% Mendelian. The older you get, the less Mendelian you are. It's important that we recognize that immunosuppressant sensitive nephrotic syndrome at this point in time is rarely reported, if ever. So when we think collectively about the patients that walk in the door, their response to steroids helps dictate whether or not they have mutations. Typically in most conditions, we don't really have common variants that have a big effect size. But in nephrotic syndromes, which are what I would say is rare complex diseases, we do have a couple of examples of that. So a GWAS of membranous nephropathy, which is another form of nephrotic syndrome, which was done back in 2011 with case sizes of 75, 146, and 335, discovered 2 loci that are genome-wide significant, mapping to the HLA locus, which is immune MHC, and PLA2R1, which is a protein expressed in the podocyte, suggesting that when together, if people are homozygous for both risk alleles, they have an 89 times increased odds of disease. So what we're really seeing here is that GWAS is uncovering a pretty biologically homogeneous condition, which is implicating immune dysregulation and a target for an antigen. This was expanded to 3,800 cases about 9 years later, which again still shows these 2 major loci, but we are identifying a small number of loci which have a smaller effect, but nonetheless, common variants, moderate to large effect sizes. APOL1 kidney disease is another disease that has been increasingly recognized as a major cause of FSGS and SRNS, particularly in people who are of West African, Sub-Saharan African ancestry, West African ancestry, who might self-report themselves as African American, Afro-Caribbean, Hispanic, etc. So there are 2 risk alleles in APOL lipoprotein L1, which are nicknamed G1 and G2. And the risk comes from having 2 alleles. And these risk alleles are common in those of African ancestry. So in the United States, individuals who would self-report as Black, about 13% of Black individuals have this high-risk genotype. And the risks of harboring this high-risk genotype are quite high. So this was originally discovered and associated with focal segmental glomerulosclerosis with an odds ratio of 17, HIV-associated nephropathy about 30. But what you can see is over the lifespan, APOL1 high-risk alleles are associated with a variety of kidney traits. And they're common because there's a heterozygous advantage that protects against sleeping sickness. And what it means is that, of all the Black patients that we see in the United States who have FSGS, about 70% of them, we can attribute their FSGS to the APOL1 high-risk genotype. And what that means is if we could figure out a cure or a drug that fights APOL1 FSGS, we have the opportunity to help a lot of people. And in fact, 2 companies, Vertex and Maze, are advancing drug therapies right now, which are small molecules inhibiting the APOL1 protein, while other groups are working on siRNAs to decrease its expression. So I'll briefly talk about this. There's one study that shows that there are low frequency variants with intermediate effects, and this was a study back in 2016 where a group led by Andre Shaw at WashU at that point, now at Genentech, looked at 178 cases of European ancestry matched with 378 controls, and they did a burden analysis of about 3,000 candidate genes for nephrotic syndrome, and they found a group of genes that collectively had multiple individuals had different predicted damaging variants in that gene, and they accumulated more in cases than controls. So you had significantly increased odds with significant p-values. This is foreshadowing to what Simone Sanakerke has led us on our RC2 group in burden analyses that we're doing a little bit more at a larger scale. I'll point out here that APOL1, which I already just told you seems to be a disease found in individuals of West African ancestry, was one of the highest odds amongst these, genotypically, genetically defined European individuals, which means, we look at this more closely on a global level. These individuals are European, but they harbor at least a chromosome 22, APOL1 in a West African haplotype. So it's really, we are challenging the orthodoxy that APOL1 is really a disease from people who say they are Black. That there are individuals who might not self-report that way. And so it gets to the idea of how we're thinking about ethnicity, race, ancestry, and trying to take the best care of our patients. And then, things that our folks are most familiar with GWAS, there's this concept of common variants with smaller effect size. And that's the story of steroid-sensitive nephrotic syndrome. So over the last decade, studies by Rashid Gabatagisan, Kazumoto Iijima, Robert Clayta, and then another one, Pierre Ronco et al., have collectively done GWAS of steroid-sensitive nephrotic syndrome with relatively small sample sizes in terms of cases in the hundreds, have collectively always found HLA as well as some other hits as well. Work led by a consortium, including again Simone, myself, Pierre in France, Kazumoto and Katsushi in Japan. We published a paper a couple of years ago that had 2,400 cases, 36,000 controls, and what we ended up finding are about 12 loci that are associated with increased odds of steroid-sensitive nephrotic syndrome. And when we did conditional meta-analyses, we found additional loci. And all of these, when you look at closest gene and do a little functional annotation, they all seem to be close to genes that are involved in immunity. And when we look in a cell-specific sort of way, they seem to be more functioning. The cell type of interest seem to be more immune cells. So this is backing up what we know already epidemiologically and clinically, that this may be more of a dysregulation of immune cells acting upon either a normal kidney or susceptible kidney. So, the next step is we need to bring this all together at scale for nephrotic syndrome. We need to get more patients, particularly from underrepresented populations. We want to identify new single gene causes of disease. We want to do more loci from GWAS. We want to empower rare variant association studies. We want to look at modifiers. We've already found a modifier of APOL1. That was a paper that came out last year led by Simone that we were able to contribute to. And we want to do it all because we're greedy and we're ambitious and our patients expect that of us. And we want to, at the same time, we recognize that data sharing is really important, and this will allow meta-analyses, bring in additional families. We've published a little bit about this, the importance of this, and it's the responsibility of all of us to facilitate this data sharing so that everyone wins. And I think there are lots of models out there, especially in other phenotypes where, PIs win, trainees win, the families win, biotech wins, and there's a way to do this without anyone getting short shrifted. So I want to just briefly end here by talking about what we've been doing to do this at scale and how we've been really appreciative of our work with the Knowledge Portal Network. So again, this is the core four. And about 5 years ago, we wrote a grant to the NIDDK to propose this really ambitious study that used a combined cohort of over 10,000 individuals with focal segmental glomerulosclerosis, steroid-resistant, steroid-responsive nephrotic syndrome patients. And what we want to do is we want to this was an RC2 study, so this was a larger than an RO1. This was a grant mechanism that's goal was for discovery, but also to create a resource for the community. So in project one, we want to discover Mendelian forms of FSGS and nephrotic syndrome, recessive and dominant. In project two, we wanted to, again, treat FSGS as a complex trait. We wanted to do association studies, modifiers, structural variants. We want to define the relationship between these two different forms of genetic architecture. And then we want to go from mapping to mechanisms through functional analysis in vitro and in vivo models. And then we want to hopefully identify small molecules. And this is a really, there's lots of ways to enter into this cycle and exit. And overall, what we want to do is turn this genetic data to clinically actionable information and share it widely. And that's how we brought in the folks from the Knowledge Portal Network, Jason, Noel, and the rest of the team into doing this through this integrated web browser. And in doing so, we brought in increasing numbers of individuals, including Drs. Rashiga Badagisan from Duke University, Opayemi Olabisi from Duke University, Moin Salim from Bristol in the UK, as well as two other cohorts. So the shared goals and the momentum from all of us coming together is really helping to empower this. So I'm going to share a couple more slides that show some of the results that we've done thus far. And thanks, Simone, for sharing this with me earlier today, so we could bring it to the larger group. So this is just a summary of the cohorts that we have. So we have cohorts led by Simone and Friedhelm and Martin, myself and Matthias Krebster and Neptune and the rest of the squad. These individuals all have either exome sequencing or genome sequencing, and ultimately we have about 5,276 cases and then 28,000 controls. And what some of the things we've done thus far, we've done a trans-ethnic nephrotic syndrome GWAS that took both adults and pediatric, both patients steroid resistant, steroid sensitive. We found HLA and we found APOL1, which gives great face validity for that we've done this correctly. And as we look more closely and as we look in subsets, we're identifying additional loci. And the important aspect of this is we are sequentially removing patients with Mendelian variants, and we're partitioning by the endophenotype, like steroid sensitive, steroid resistant, that are allowing us to have a cleaner GWAS, because these rare cases of Mendelian disease still impact our GWAS more than if we had like hundreds of thousands of cases. And we're finding immune drivers of immune and non-immune. So this is what happens when we look at only steroid-sensitive nephrotic syndrome. This was from our paper a couple years ago, which highlights, interestingly, a lot of immune genes, but also NPHS1, which is a fundamental podocyte gene, and it's a Mendelian form of steroid-resistant nephrotic syndrome. And it raises the idea that has been seen in other conditions, including kidney disease, that a single gene with rare, highly deleterious changes can cause a Mendelian disease, but common variants, whether it's exonic or regulatory, can lead to a more common complex trait. And nephrin has been hot these days because recently in minimal change disease and other forms of nephrotic syndrome, we found antibodies to nephrin. So there's a lot of work that's being done, that's being uncovered by large-scale GWAS. This is really driven by Japanese and East Asian individuals. So that is showing us the importance of global recruitment of patients. So it's all coming together around this. And then finally, work done by Simone here, really led by Simone with all of our patients. We're doing exome-wide collapsing studies, burden analyses. This is under a dominant model for protein truncating variants. So this is, PTVs only is the most stringent. This is when you do dominant models with protein truncating variants and missense alleles, and this is under a recessive model. And what we're showing here is, the QQ plot, 95% confidence intervals. Each dot is a gene here. And what's really cool is that most of the genes that are popping up across these models are genes that have been discovered through family-based analyses. And it's also revealing some novel genes that haven't been discovered in this way, but it's giving us hope that the more we can do this, aggregate cases and smartly correct for population stratification, match cases and controls, that doing non-family-based rare variant analysis should uncover novel genes, which should ultimately get us to mechanisms and then medicines. So with that, I'm going to stop here because the next step here is for Mackenzie and the group to show how we are trying to integrate this with their team to broadly disseminate that. So I'll kick it over to you.
Great, thank you, Matt. I think that's a great intro into what I'm going to be showing now, which is our beta site for the Nephrotic Syndrome Knowledge Portal. Can everyone see my screen okay? Is this showing fine, Matt?
Yes.
Okay, perfect. Thank you. And so essentially, in our project, we worked strongly with Matt and colleagues to determine what a community portal would look like for nephrotic disease. And so with that, here, this is the landing page. And as you can see here, we have a single search at the top. And this is going to be one of the key features as we walk through this. But as I'm still on the homepage, I just want to show everyone what else is on here as well. So we have information about the portal. So if you want to look into the cohorts and the data, you have the opportunity to do that. There's some other information about the project, specifically the various labs that have contributed, and all of these blue items link out to their specific pages as well. So anytime you see a blue link on a page, it will take you outside of this portal itself, or will take you to another page. And then at the bottom here, we have our other knowledge portals that are in the network and that are related to nephrotic disease. And then we also have information for how to stay in touch with us and other related resources that we are eager to connect with. And then at the top, we have our menu bar. So here, again, if you want to explore the data, you can look at the cohorts, and then there's other information about the portal itself. So I'm going to start us off with searching for a gene of interest. And at the bottom here, you can see examples of genes that are important for research. So I'm going to go ahead and search for NPHS1. And as I'm doing this, it'll take you to the gene page, which has information specific to the gene that you researched. So the first thing that you can see on the top of this page is the variants in the NephKP database itself. So here, this table displays aggregate allele counts for the cohorts. And as you can see here, they are ranked by impact, with high impact being red, moderate being yellow, green being low, and modifier as this gray tone. And this color you'll be able to see here in the consequence. So you can rank by impact if you're like, but we're just going to keep it in this format for now. And so in this table, you can see on the left the variants that come up. And so here, like I said, because this is blue, this will take you to that specific variant, which I will go back and show you more about this in a few minutes. But as you can see here, we have the allele counts, so the count, number, frequency, and homozygotes, and then there's also the nomad allele. So we have information from nomad. And if you are curious to see additional data, we have two options for subtables in this specific variant table. So the first is to show the phenotypes. So when you click here, you'll be able to see all of the phenotypes. And then these are ranked by allele number, as you can see here. And then if you are interested, you could also look at the annotations. And so I'll just show you an example here. If you were to click here on this feature, this will link out specifically to that related page as you can see here, in Ensemble. And so now we can go back to our NephKP, but if you're interested, you can stay on an Ensemble and look a little bit further. Underneath our variant table, we also have the functional associations for NPHS1, which is the gene that we searched. So here, this gives a description of the function, and here this tells us that this gene plays a role in the development or function of the kidney glomerular filtration barrier. And you have some information on here as well. As we tab over, we have gene ontology or GO annotations. We also have pathways, which are pulled in from Reactome, KEGG, BioCarta, and Wiki Pathways. Again, all of these blue links will take you out to the various resources, and the source is listed here for the information of where it's being pulled from. And then we also have the UniProt cross-references. And then, as I mentioned, Ensemble before, we also have this external resource here and Open Targets Genetics. as I go back up here, one thing that I do want to share that I didn't mention before is all of these tables are paginated. So at the bottom, if you're looking for more information, you can use the various arrows to explore further in. We just do this so that you can see a shorter summary for, quick, easy reference of information. So as I said before, in this variant table, so we have this variant here, and let's say I'm interested in learning more about what this variant has to offer. So if I click on the variant itself, it will take you to the variant page in the NephKP. There is a lot of data, so it might take a minute to load, but now you can see here, this is the variant that is related to the gene that we searched. Now, if you want to go and search for a different variant, you could input it in the search bar at the top here. And again, these are other examples. I'm actually going to go back to the home page quickly just to show you that you can also look for this in the search box. So I'm just going to copy in this one here. And so I just want to show you that you can search from the home page as well. So there's various different ways to get to the variant or gene that you're interested in. So for this variant here, you can see the first thing on the variant page are the phenotypes of variant carriers in NephKP. So this was a part of the subtable that I had just shown you in the gene page, but this gives you more of an opportunity to explore the data. So as you can see here, we have the phenotype, allele count, allele number, homozygotes, and then allele frequency. And you can explore all the information by either all the samples or broken down into specific phenotypes. So below this table, we also have the predicted variant consequences. Again, this has the same labeling for impact. So if you're interested, you can take a look here. And again, there are multiple pages. So if you're eager to explore further than this, you can. And then at the bottom here, we have the Nomad population frequencies, which are pulled in from the Nomad browser. So let's say, again, you're eager to see more from what Nomad has to offer. You could click View in Genome browser, and I guess this page doesn't exist, but there are other ones that will connect to Nomad itself. One other thing that I want to mention is for any of the data with any of the tables, you can also download this data and you can save it as CSV, TSV, or a JSON file, depending on how you would like to use the data. So these are the main two pages that we have. The one thing that I want to share with you is the cohort page as well. So Matt walked through, who would be contributing to this database, but from here, these are the samples that we have currently in the NephKP. So as this portal expands and grows, we're hoping that we'll be able to have this as a community resource. So other individuals will be able to input data, and we'll be able to have more information as we go. If you're curious about how the data was generated, so we have a summary here of all the information, and then we also have detailed information for the analysis, so specifically for the variant calling and quality control. So if you want to take a look further into this, these are just the brief summaries, but you can also take a look at the information regarding this specifically. And so one other thing that I just want to share too is there's a QC report that you can download as well if you're interested in that for the data. Just heading back to the home page, as I said, this is a community portal. It is in the beta stage, so we are really excited about what we've been able to put together so far, and we're hoping that this becomes more of a community resource for people who study nephrotic syndrome and for others that might be interested in using this data. We're hoping that as part of this, we can expand upon it and have more members of the community use this in their day-to-day research. As we had mentioned before, that there are the related resources here. but we're eager to include more features from the various resources, have included other data and update on some of the features so that we can best represent the data as they're available in the Knowledge Portal. I did see there was a question in the chat, so I'm just going to go to that quickly. Oh, okay, that wasn't a question for this. All right. So this is a brief overview, and I know I went through this pretty quickly, but I would love to have the opportunity for people to give us feedback specifically on maybe something that you're interested about doing for research and seeing how this can help you, or if you have any feedback or have any recommendations, we're eager to hear from you as well. This is an opportunity to hear from the group. And so again, we just want to make sure that this resource is, the best suited for the community that will be using it. So there, I'll pause for a moment and see if anyone has any feedback or if they have any questions about the site. I do know that I went through pretty quick.
Thank you, Mackenzie and Matt. That was fantastic. I guess while we're waiting for feedback to trickle in, I have a couple questions for Matt about the scientific portion. I really loved your approach for complementary modeling, I guess. So using, say, fish, cell lines, mice to model some of these. So in your experience so far, which models have actually proven most useful? Do you find differences in the validity of different models for, say, the more common GWAS variants versus the modifiers versus the more highly penetrant Mendelian forms?
Yeah, thanks for the question. And I'll start answering, and then Simone, if you're in a position to also contribute, please feel free, okay? So I think the history of modeling for genetic variants in nephrotic syndrome has been mainly limited to Mendelian forms of the disease, and in that case, mice have really been the mainstay. People have not really extensively done a lot of workup of genetic variants from GWAS. Some work has been done in zebrafish, fruit flies. Amazingly, a lot of their fundamental cells that wrap around the heart have a lot of homology with Mendelian genes. So that's been a tool as well. Simone, anything from your side?
No, not much to add. Yeah, so not much modeling from GWAS, also because we don't have a lot of GWAS for nephrotic syndrome. We only have the one for steroid sensitive, nothing from structural and the FSGS. Yeah.
And one thing I would say moving forward is that, just like many fields, organoids are starting to become higher fidelity to represent different organs. And in nephrology, it's the same. Interestingly, the kidney organoids, the most mature, the most far advanced part of the kidney is the podocyte. So on one hand, the podocyte gives us the opportunity to study podocytes in organoid systems. At the same time, they still do not represent a fully formed podocyte. But what I would say is, moving forward, I think a lot of our colleagues are working on iPSC-derived organoids and glomeruli on chip, which should give us an avenue in if Simone and I and the rest of us can find targets to evaluate.
Nice. That's really exciting.
I did see there was a comment in the chat about the link not working. I just sent the beta site link, so hopefully that should work. Like I said, this is a beta site, so it's not on our network yet. So it is, in the early stages. So hopefully the link should work though, and everyone can take a look and explore for themselves.
I guess while folks are clicking around, I have a couple other questions too. So I liked what you showed with the rare variant exome-wide collapsing. I thought it was interesting that some of the genes showed up among multiple models, right? So you would see, say, col, what was it, 4A3 or something under the dominant PTV and also the recessive model. If you look at the individuals that have those variants, do you see that, say, it's a hypomorphic variant under the recessive model versus like haploinsufficiency and a protein truncating variant under the dominant model? Or do you see differences in the endophenotypes of those individuals?
No, not really. It's just a matter of how the dominant and recessive models are built. So recessive genes, if they're very prevalent, they will show up in the dominant model as well because they have at least one variant, right? The
Got
dominant
it.
is,
Okay.
yeah.
Yeah.
So when the gene is very strongly uh, associated or prevalent in your population is recessive, you will often see show up in the dominant model. And that's also important to recognize it because these tests, especially for discovery, they don't allow you univocally to make a statement on what's going to be the real mode of inheritance, right? So that's the thing. So if it shows up in recessive only, then you're kind of sure that it's recessive genes. If it shows up in the dominant, it could still be a recessive gene.
That makes sense. Thanks.
Any other questions for people? So if you have any questions, please feel free to use the chat Q&A or raise your hand and I'll allow you to talk if you're interested in asking in that mechanism.
Yes, one other question is we're thinking we have some folks who are on from some of our other community portals, right? And one thing that we're always thinking about is do some of these phenotypes, say, have overlap with some of the other phenotypes in these other portals? And over time, could we form connections there? As we're looking at some of the variants that you showed, which are, high, more common sort of, GWAS SNPs, are any of those loci associated with other common phenotypes or immune phenotypes that you're aware of?
Yeah, absolutely. I think you're right on point. People have done formal genetic correlation studies to find overlaps of, some of our kidney GWAS with other kidney traits, but also other immune dysregulation, white blood cell quantitative traits. This is mostly for steroid-sensitive nephrotic syndrome, membranous nephropathy, IgA nephropathy. But, ultimately, it'll be a great thing to holistically view these together, both formally in terms of statistical models, but in terms of lookups as well, to see. And, I know that Jason and, part of his research is really focused on kind of doing this. So, in the future, we'll discover a lot from doing that. Simone, anything to add for that?
Yeah, just the formal genetic testing for correlation, I think is going to be feasible in terms of with one limitation though that we have with some of the studies, and probably also with other discovery studies, that these are not EHR-based studies, so the level of clinical information that we have is limited, and is usually focused, like zoom focused on the disease phenotype because they were ascertained for having that phenotype because of the genetic study. So we oftentimes, if you find variants that we believe they might explain additional phenotypes, we have to go back and recharacterize like as a reverse phenotyping type of strategy to look at correlated phenotypes. But we don't have that at hand.
Yeah, and one
Almost
thing
never.
that we've talked about too is potentially adding a feature on the knowledge portal that we'd be able to see, where those connections are made, whether that be in a table or some other type of feature. I know that is something that people have said could be useful, and it's been mentioned a couple times. So I think if this is something that more people are interested in, it could add value specifically to our NFKP. Any other questions from the participants? Okay, I'm not seeing any, but Matt, is there anything else you want to add, or Simone, before we wrap up?
No, I mean, I think that, for many, many decades, discovery has stayed within one person's lab, and more recently in genetics and nephrology, we're really heartened for to collaborate together. And I think this represents the next step to more broadly disseminate it. And my hope is that this will end up being widely as impactful as possible. And thank you guys for empowering it. And it was great to join you. And we'll be good to get a little lunch before the 1 o'clock meetings start.
Great. Well, thank you so much to have your presentation, Matt and Simone, for joining. And thank you to everyone for participating. Just a reminder that our next session is going to be May 8th. It'll be a focus group meeting. I will send more information about that once we get closer to. But thank you to everyone. And if your colleagues weren't able to join, please feel free to share this recording afterwards. So thank you again, everyone.
Thank you. Bye.
Thank you.
Bye.
Bye-bye.